Hi, my name is Linnea.
I'm Bezia.
And I'm Linnea, and this is episode number 23 of the Lemma Journal Club.
Is that right?
I don't think so.
Shit.
Unless you've already changed it.
All right, let's keep going, but I assume that it's episode 24 of the Lemma Journal Club. Linnea, what is this algae paper?
The Lemma Journal Club is the show where we read every paper to tell you if you need to read it. You probably do.
Every algae paper.
We have a terrific paper for you today. We are talking about the research paper, The Life Cycle of a Cyanobacterial Carboxysome.
This is my line. It's in Science Advances by Nicholas Hill, Jean White, Sabina Altus, David Bortz, and Jeffrey Cameron.
Great.
Published 2020. Linnea, why are we doing another algae paper?
This isn't an algae paper.
It's an algae paper.
This is a cyanobacteria paper.
If this thing blooms, do you call it an algal bloom?
The nomenclature, I mean like the lay people, algal bloom.
So this is not an algae paper in the sense that like chickens are not reptiles.
This is a cyanobacterial paper.
This paper very early on says cyanobacteria perform more than 35% of global CO2 fixation, despite the fact that they comprise less than 0.2 of photosynthetic biomass.
Yeah, I highlighted this in red because I was unsure if this was true.
Honorific algae. That's what I'm saying.
Sure, yeah. That seems fair.
If we sound different today, we are recording from APS March meeting. We are just on a improv recording setup, so we'll see how it goes.
We've never used these microphones before, so...
We found out a couple hours ago that our flight home is canceled. There was some idea of getting home, recording this, and then putting it out in the nick of time, but that's not going to happen, so we're going to record it far away. This was, I think you're going to have to do most of the talking for this paper.
So have you already asked me why I picked it?
You picked it because it's an algae paper.
No, I didn't.
All right, Linnea, why did you pick this algae paper?
I picked this because it was an algae paper. No, I mean, partially, I love the carboxysome because it's kind of like a pyrenoid, but it's teeny tiny. And
is it smaller?
Yes, teeny tiny.
All right, so then I'm going to interrupt you right with my chief complaint about this paper.
Oh.
There's no scale or time anywhere.
Oh, yeah?
There are two scale bars in the paper. In B and C, there's a scale bar.
Yeah.
But there's no text that tells you what that scale bar is. And there's no other scale bars in the rest of the paper. There's no scale bars in the movie. There's no timestamps in the movie.
Okay.
There's no text anywhere that would tell you how big anything is.
Okay. That's fair. I know a carboxysome is teeny tiny.
But how teeny tiny?
I think they said that somewhere this was like each one is a micron long.
That actually does answer a lot of my questions though, because the reason why I was annoyed by the scale bars is I was like, these pictures aren't very good. Are these things really small?
Oh yeah, they're really small, which is why they're like not as fun to study as pyrenoids. But they do a lot of CO2 fixation.
Do?
They do,
Mm-hmm.
yeah. They do a lot of CO2 fixation. And I was talking to some people at Carnegie Mellon about mother machines. And yeah, I was thinking about how do carboxysomes get inherited and could a mother machine help us think about that? And I thought this paper would give me a sense of what is known in the field already or was known in 2020. So I went to a conference. There's a big conference of all of the people who study carbon concentrating mechanisms in both eukaryotic algae, which is what I call algae, and cyanobacteria. And so I heard a little bit about these cyanobacterial CCMs, carboxysomes, at the last CCM meeting I attended, which was a long time ago, 2022. And so this is kind of where I wanted to read the paper that was the talk that I watched.
So they mess with this bacteria. That's what it is, really? It's a bacteria?
Yeah, it's a photosynthetic bacteria.
They mess with this bacteria so that it will make its carboxysomes in response to IPTG exclusively.
Yeah.
And then they mess with whether or not they're giving it IPTG. And then they split the bacteria into, according to their text, into 4 categories, which is, quote, too short, no growth, growth, and degradation.
Yeah.
But according to all of their figures is growth, growth prime, degradation, no growth. And there's a disconnect between the text and the figures that doesn't seem to get resolved ever. There's a couple times that the text has some weird things going on. They start talking about referring to some of the carboxysomes as non-aging carboxysomes. They just like pull the word aging out of, like, we're definitely not talking about aging in any way whatsoever here.
What do you mean? I think that this could be a way to study molecular aging.
Absolutely not.
What do you mean?
What do they mean? It's just whether or not it's growing or not.
Yeah, but so why does the carboxysome get, how does it go bad? Why does it get tagged for degradation? Why can't you just make one carboxysome and have it forever? This is a molecular aging question.
I don't know.
Do you know what a carboxysome is?
Yeah.
Okay.
Maybe. It's a pyrenoid light.
Yeah, it's also kind of like a virus capsid.
They
look
claim
very
this.
similarly.
That it has a shell.
Yeah.
How well is it known that it has a shell?
Very well.
Okay, then they, with absolutely no evidence, say that it degrades because the shell was broken and thus it degrades.
I think they don't have any evidence for that.
They say that so many times.
But they never say they have any evidence for it.
Well, they clearly just got hit by the reviewers, where the reviewers were like, "You have no evidence for this." But they even in the discussion where they're trying to backtrack, they're like, "Our results suggest that shell breakage may be the dominant mechanism of carboxysome activity loss." And then
Highly suggestive.
They just say our results suggest that.
Oh, they say somewhere in the discussion, the like pole migration before disappearing completely, highly suggestive of a carboxysome shell breakage event.
Yeah. They have absolutely really no reason to think that. And they just walk it back very slightly for the reviewers. We were unable to show that shell breakage is directly responsible for carboxysome activity loss. You haven't shown anything about a shell. I don't see a shell. I see a small glowing blue dot.
Yeah. Yeah, here, I mean, again, because they're teeny tiny, yeah, it's hard to, like, even if they were able, I think they said in the discussion that they wanted to, like, co-label the Rubisco and the shell.
I think that was the original. That would be the
definitive experiment.
Yeah.
But even then, it's, like, such a small thing.
It seems to me like the point of this paper was supposed to be that they were going to study that. They didn't successfully study that. And so they just kind of have this, here's some stuff paper.
Yeah, it definitely wasn't exactly what I wanted it to be. I wanted it to be, you trap one cell, Merriam style, and let that divide and see how the carboxysomes go out, and not get one carboxysome and see how long it can.
But to answer your question, no, I do not, like, it's interesting because we talk about like Jordy's aging ribosome thing. And there I'm okay with using the word aging. I don't really have a strong sense in my mind if that has anything to do with what we mean by like humans aging or
right.
animals
I think that's
aging.
a big question mark, right?
Right. But in general, when I'm like, I don't really want to talk about these plastids malfunctioning or organelles malfunctioning as being the similar thing to aging. I think that's like maybe crossing wires a little bit. And so I do not want to talk about this carboxysome being hit for degradation as these things aging. Because aging means something to me, right?
But like then you don't want to talk about ribosomes aging.
Right. And that's what I was getting at, is that we've entered an interesting, like, because I have never had any problem with Jordy talking about ribosomes aging, but it is a good point that we're mixing up nomenclature here.
Yeah.
And we're probably doing that for funding reasons, that it's much easier to get funding if people think that you're studying, like, humans aging.
Yeah.
Right?
And for ribosomes, it's like, maybe. Yeah. But for carboxysomes, it's like,
clearly,
what are you talking about aging in a bacteria? Like this thing lives, it doesn't age, right?
It's an interesting hit to the ribosome. It's tricky, right? Because like this kind of stuff certainly might have something to do with aging. But to say that having an organelle marked for degradation is the same as the cell that has that, I mean, it could be like the reverse of aging, right? It could be euthanizing, or I guess euthanizing has a completely different meaning.
Yeah, I mean, like an aged cell might be worse. I mean, this is part of Jordy's paper, right? Is like an aged cell might be worse at recognizing that a protein has degraded in the cell, has been around for too long and gained too many modifications, and is no longer functioning. And so like a healthy cell is able to say, oh, this is a bad thing, let's get rid of it. And a aged, quote unquote, in like chronological time cell might not do that so well. But here I think they're just thinking about like the molecular function. How long can this thing be functional, right? I was a little upset that they didn't have that many that they looked at. They looked at 28 carboxysome trees. And they had one super carboxysome.
Sniff. Every sniff is going to be a whole edit that I've got to make.
Okay, I'll stop sniffing. I'll just be covered with
dog
mucus.
We're in a very dog-heavy Airbnb that...
I feel like I'm getting more allergic to dogs the longer I've been married to you.
If you could add one experiment to this paper, what would you add?
The mother machine.
The mother machine. You want to explain to people what a mother machine is?
A mother machine is a microfluidic device where you trap a single bacteria in a channel, and then it can't move, but it can divide. And so it creates a bunch of progeny, but it's stuck in its little channel. Is that a good explanation?
Sure. You asked me to do an experiment with carboxysomes in a mother machine because you felt like I had nothing to do. Yeah. What was the experiment you wanted me to do?
Yeah, I want to label carboxysomes.
With what?
With GFP or whatever.
Okay.
And yeah, if you have the bacteria trapped, the mother bacteria trapped, and it starts dividing, like at what rate are you getting new carboxysomes? At what rate are those carboxysomes dying, like during these divisions? Can you track a single carboxysome and see how many life cycles it exists for? Like, what's the average age of a carboxysome?
I have a great comment here. My comments are mostly, I just keep highlighting things in red. It just says, huh? Huh?
Sorry, I didn't realize it would be such a dud on your end.
I think I just came into it with a negative psychology. I came into it's Science Advances, which
you just don't like the journal.
Well, it's just where my paper is going.
Yeah.
And I'm really feel like I don't know how I will ever write a paper better than this paper I just wrote.
Oh.
And if this is the best I could possibly do.
No, you'll write a better paper.
I don't know. It's really good. And
it's a really good paper. But I mean, just being at this March meeting and talking, we're at this weird stage in our career, right? Where all of our friends are professors.
And we're the only ones who aren't.
And so everyone I've talked to at this meeting has been a professor, and they all have groups that they're running, and they're doing they're thinking on a different scale than we get to think. And so you'll write a better paper just because of that.
But also these are all UC Boulder people. And we started this week by looking up why UC Boulder never hired us.
I didn't know it was from UC Boulder.
You don't know Jeffrey Cameron at UC Boulder?
I don't.
Okay. Yeah.
But maybe we should. If they're Department of Biochemistry.
Yeah, you could ask them
Renewable
to do
and Sustainable Energy Institute.
Your mother machine experiment. Somebody's
in the Biofrontiers Institute. Yeah, I mean, this experiment was very up the alley of this Feng-Wei C, who I talked to at Carnegie Mellon, who's very interested in, how the physiology of cells changes with.
But I'm happy that I learned about the carboxysome. You mention this word every once in a while, and it's good for me to sat there and looked up how different things work in it. And I spend a little bit on this time on this CCM operon and understanding how they've got this IPTG
inducible system.
Yeah. And I was happy that I did that.
Yeah.
I originally just put that all in red. I was like, this is too dense. And then I was like, no, I'm going to actually learn what they're talking about here.
Yeah, I mean, it's a neat thing that you can do is to induce carboxysomes and then turn them off. And it's not an experiment I would have thought of.
Yeah, and I guess that's the beautiful thing that did work for them was this inducible CCM, and that's the beauty that we're supposed to take away.
Yeah, it does have some movies.
Yeah, thank God. So we'll use those movies.
And it highlights the super carboxysome that was able to support 5 life cycles or 5 cell divisions.
Why did they highlight that? So this is One remarkable carboxysome reached a net productivity of 18 microns. You love this? In less than 50 hours.
I thought it was hilarious.
Single-handedly supporting 7 generations of growth.
I thought it was like an advertisement for
this one carboxysome.
It's like a rec letter for this carboxysome.
Like, what the hell is this? We can't put any of this out. I can't say any of this. We'll get absolutely roasted by everyone.
It's okay. Nobody is listening. Although I guess post-March meeting, maybe people will listen to this one episode because they just saw us.
I doubt it. I do wonder what actually happened for some of the text. Some of the text doesn't meet the figures.
Yeah.
Which is bad.
I did have trouble with the text figure coherence, and I dealt with it by reading the text and then reading the figures as like two separate entities. Which tell kind of different stories, but...
Yeah. For the most part, like, I think it's fine, but I don't have much to say about it. It doesn't have much to say about it.
Okay. Yeah. I mean, I think the thing that I find intriguing about the paper is, thinking about molecular... What do you want to call it if not aging?
Yeah. It's rough, because I want to call it molecular aging. I like that term. The problem is that I think...
the connotations are not right.
Yeah, that you're just fooling people into funding you because they think that you're studying like mammalian aging, and that feels pretty slimy to me.
But I think Jordy has really sold me on this being an interesting question, and this is an interesting way to study it.
Sure.
And I think if you got way more statistics,
I think I just want to put the word molecular in front every single time.
Every single time.
Single time. Never say aging. Always say molecular aging until someone shows a strong link between the two.
Yeah.
And even then, still keep saying molecular aging, because it's sometimes going to be a link and sometimes there's not.
Yeah. I still think it's not the greatest, but I don't have a better word because it's still, the connotations are still there. But I think the thing I found beautiful about it was like, this is a system where you can like try and track. And I think I'm trying to think of an experiment that you could do like more precisely to track could you track a single carboxysome in a fully functional cell that has all of its other carboxysomes also active, and see how long that guy sticks around.
There's no way to know because there's no scale bar. So it could be 10 nanometers, it could be 10 microns.
Oh, you definitely can see individual carboxysomes.
What do you think the scale bar in 1B is supposed to be?
I don't know. A micron? Half a micron? They really don't say?
No, I couldn't find, I did like a control F for micron or or micrometer or any of these things.
Well, let's look up what the size of our carboxysome is. I think it's like 500 nanometers, but carboxysome.
I just feel like it could have been in the paper.
Size. Yeah, that would be good.
The only thing that they have is in figure 4.
Carboxysomes are teeny tiny.
They have
160 nanometers.
Kind of a scale bar in figure 4 that you can kind of back things out of, but it's,
Because of this chymograph?
Yeah, and you can implies that like one of these cells is like roughly half a micron long, and then the carboxysome is roughly like 100 nanometers in diameter.
Wikipedia says 100 nanometers.
Yeah.
So small, but they're not like on top of each other. They're usually spaced out from TEMs, so you could track them individually.
Do you have more important things to say?
More important things to say?
About the paper?
I don't think so.
I think it's a good discussion. Like, it's not a super big paper. We don't have to drag this out for an hour. It's okay to have a short episode every once in a while.
I highlighted 17 and 18 as papers I wanted to read.
I have a pretty long list all of a sudden of papers that I want to read. Some of them from this conversation. Yeah, this spatially ordered dynamics of the bacterial carbon.
Yeah.
Although it's pretty old, so maybe there's newer stuff.
It must be how this Savage was the last author on one paper we did before, which was the Rubisco mutations paper. So maybe this is the paper that got him his job. Yeah,
I have a bunch of stuff. I want to do a Morel paper and do it in depth and really think hard about it.
Okay.
Because we've been talking about it a lot, and it's been a long time since I've thought about one of his papers in depth. And maybe actually we'll want to like ask someone like Danny to hop in on that with us.
Sure. So much reading to be done. And one thing I'm really terrified of, which I don't know if we want to include in the podcast, but we can delete it, is that if we leave science, we're done. We have a finite. We could have a finite number of podcasts. And a finite number of papers. And so, why am I wasting my picks on these?
On the life cycle of a cyanobacterial carboxysome, because you tried to do a cyanobacteria carboxysome paper, and it's hard to know where to start. Right. And so this is where you started. Maybe we'll do something a little bit better.
But it's just like I should have done more homework if this is like the,
But
you
then
know,
you
if we
get
only have
homework
10 more.
paralysis,
Yeah,
right?
yeah.
Like, so.
But yeah, interesting paper. And I think, yeah, it was sparked by these conversations I had at CMU. So that's always nice to be curious about other people's work.